This is the video for experiment 3, determine number of microorganisms in a sample. For this experiment, there are general methods to estimate the microbes populations, which is the first one, direct count of cell number under the microscope or by an electronic particles counter. And the second one also a direct measurement of microbes biomass by weighing whole cell or refer to the standard curve. Besides direct method to estimate the microbes populations, it also provides two indirectly method to estimate the microbes populations, which is the third and the first indirectly count of cell, which estimate the number of microorganisms in the original sample by the formations of colony on a agar plate. And the other indirectly method is indirectly estimate of microbes biomass by measuring relative constant biochemical component of microbes cell or refer to the standard curve of microbes. For this experiment, there are two objectives introduced, which is the first one to determine the number of microorganisms in a sample based on the process of enumerations, and the second one is able to understand the several methods for enumerating microorganisms. In this experiment, there are two methods involved for determining number of microorganisms in a sample, which is the pour plate method and the split plate method. The general definitions for pour plate method is a method where plating of higher volume of sample by dispensing a liquid sample onto an empty pretty dish, which is then filled up with a molten medium such as typic soy agar or known as TSA, or a selective medium such as violent red blue glucose agar or short form known as VRBG as well. The next one, split plate method. The definitions for split plate method, it is a method for the isolations and enumerations of microorganisms by pipette the sample on the agar medium and split this sample all over the surface of agar by using an sterilized glass roll.
The procedure began by melting 6 nutrient agar pores in a boiling water bath, then placing them in a 50 degrees Celsius water bath. 3 dilution blanks, which were 10 to the power -2, -4, and -6, and 6 battery plates, which were 10 to the power -2 through 10 to the power -7, were labeled. The unknown sample was shaken and 1 ml was transferred to the 10 to the power -2 dilution blank, which was vigorously shaken. From the 10 to the power -2 dilution, 0.1 ml and 1 ml were transferred to the agar plates labeled 10 to the power -3 and -2 respectively. Another 1 ml was moved to the 10 to the power -4 dilution blank. This process was repeated for 10 to the power -5 and -6 dilution blanks. Melted agar was poured into each battery plate with sample dilution mixed and allowed to cool. Inverted plates were incubated at 30 degrees Celsius, and after incubation, colonies were counted, marked, and recorded. Then, plates with over 300 colonies were noted as the NTC, and the concentration of bacteria in the original sample was calculated using plate count data, considering plates with 30 to 300 colonies, where each colony forming unit CFU represented the progeny of a single cell. For example, if 200 colonies were counted on the 10 to the power -4 plate, the original sample had 2 times 10 to the power 6 viable cells per ml. 3 9 dilution water blanks were labeled 10 to the power -1, -2, and -3, and 4 nutrient agar plates were also labeled 10 to the power -1 through 10 to the power -4. A 1 ml water sample was aseptically pipetted into the 10 to the power -1 dilution tube, and the tube was truthfully mixed by vortexing. Using a new pipette, 1 ml was transferred from the 10 to the power -1 tube to the 10 to the power -2 tube, and the process was repeated for the 10 to the power -3 dilution. Following the same procedure, 1 ml from the 10 to the power negative 2 dilution was transferred to the 10 to the power negative 3 dilution tube, creating a concentration of 1 per 1000 of the original sample. Using a new sterile pipette, 0.1 ml from each dilution tube and the original sample was transferred to agar plates. This step effectively dilute the sample by another factor of 10, resulting in plates ranging from 1 per 10 to 1 per 10,000 of the original concentration. After sterilizing a glass spreader by dipping it in alcohol and flaming it in a Bunsen burner flame, the glass rod was allowed to cool. The sample was then sprayed over the surface of each plate by touching the rod to the agar and rotating the plate. This spreading procedure was repeated for each plate, ensuring sterilization and cooling of the glass rod before spreading each sample. Finally, the plates were incubated inverted at 30 degrees Celsius for 24 to 48 hours. Following incubation, colonies were counted, focusing on plates with a colony range of 30 to 300. Colony counts were recorded and concentrations of bacteria in the original sample were calculated based on the dilution factors.
This section is the overview result that obtained from the experiment, which is the right-hand side represent the split plate method, and the left-hand side represent the result of pour plate method. Based on the diagram, there is observations that made from split plate method, which is the mark on the pretty dish. It shows the number of colonies formed from a sample. For the pour plate method, some of the pretty dish show the colony of microorganisms and some of didn't. And the reason why the colonies does not form on the agar medium will be explained on the discussion part.
Why are counts above 300 and below 30 statistically unreliable? The plates with counts less than 30 colonies are not statistically significant as they are too few to be representative of the sample. Meanwhile, counts with greater than 300 colonies are not considered valid because they are likely to produce colonies too close to each other to be distinguished as distinct colony-forming units. Did you enumerate all the bacteria using this method? The answer to that is no, as the temperature of the agar might kill or damage heat-sensitive bacteria, as not all the bacteria can tolerate exposure to a temperature of 45 degrees Celsius. Thus, some of the bacteria was not counted accurately, as some of the bacteria might be killed during the process. What factors make the pour plate method selective? And how can you reduce the selectivity of this procedure? The high temperature of the molten agar affects the selectivity of the pour plate method. This is because a hot molten agar would kill or damage heat-sensitive bacteria. To reduce the selectivity of this procedure, the liquefied agar is left to cool but not solidify, before being poured into a sterile petri dish. How can you achieve a 1/100 dilution other than by adding 1 ml of sample to a 99 ml dilution blank? This can be achieved by adding 0.1 ml of a sample to a 9 ml dilution blank, or adding 1 ml sample from a 1 out of 10 dilution to a 9 ml dilution blank. What does the term colony forming unit mean, and how does it differ from a colony? The colony forming unit CFU is a measure of viable clonogenic cell numbers in the form of CFU-ml. It's also used in microbiology to estimate the number of viable bacteria or fungi in a sample. A colony can be made up of few microbes that are not visible under the naked eye. But colony-forming units is a colony of bacteria that is visible under the naked eye and can be produced by several colonies. What types of errors would be introduced by using a single pipette for all operations in this procedure? Systematic errors would be introduced if a single pipette is used for all operations in this procedure. What are the advantages and limitations of the spread plate method compared to other enumeration procedures? The spread plate procedure is suitable to isolate colonies for further analysis because colonies grow accessibly on the agar surface, whereas the colonies become embedded in the agar when using the pour plate procedure. Besides that, unlike the pour plate method, the spread plate method is suitable to handle heat-sensitive cultures. However, the spread plate method is not selective and may allow for the growth of other microbes and the presence of more colony-forming units. In addition, it does not allow for the growth of obligate anaerobic microorganisms. Next, how could you modify the spread plate procedure to enumerate obligate anaerobes? We can use an anaerobic jar after inoculation to enumerate obligate anaerobes because anaerobic jars will create an anaerobic environment for obligate anaerobes.